I'm going to be telling you about some work that I've done with Michael Newbert and Heidi Sossack's lab at Woods Hole Oceanographic Institution. On this title slide is a photo from one of the cruises I'll be talking about. It's a picture that I took on my phone in panorama mode. So the idea is it takes a series of pictures and then merges them together. But as you can probably tell, the ship was taking some waves, and we end up with a wobbly horizon and some discontinuity here. And I've included it because I think this captures some of what I'm trying to do with this work, which is look at continuous observations from a moving vessel in a moving environment and piece them together to create a picture that captures the dynamics of this system. So just keep that in mind. I'll start by introducing the main characters. These are the Picophytoplankton. This is a group that's classified by their cell traits. They're photosynthetic, and they're relatively small, 1 to 2 microns in diameter. And we've heard about some of these groups today, or in this week, Prochlorococcus, and also the Pico-eukaryotes. I'll be talking mostly about Synechococcus. You can see the distribution on the right. So it's very abundant all over the world, concentrations of tens of thousands of cells per milliliter. And so this group is contributing greatly to global primary production. We're also expecting the Picophytoplankton to make up a larger component of the phytoplankton community as the climate changes. So we're really interested in the role that these organisms play in their ecosystem and how that might change over time and with changing environmental conditions. So over the last week, I've inferred that this is a trait conference. And so I'm going to highlight the ways that traits come into this work. I've already mentioned that they come in for our definitions. The Picophytoplankton are defined based on their morphological and physiological traits. I'll be using flow cytometry measurements, which are measuring cell size and fluorescence. We'll also be estimating growth rate, which is a trait. And we'll be trying to make predictions about the responses to environmental change, which some of us here would argue is a trait as well. So this is the goal that I'm after. Question being, how do the Picophytoplankton respond to environmental variability? And I'm not particularly interested in how one species responds in a lab or in an experiment, but how the community responds in situ. And we're not going to use a crystal ball to do that. All we need is observations of the Picophytoplankton across a variety of environmental conditions. We've done that in 2 ways. The first being looking at a stationary observatory and watching as the environment changes over time. And the second, going off and observing over space across the spatial gradient in environmental conditions. And this is going to be the structure of the rest of this talk. I'll start by going over what we have learned from the stationary observations, and then moving on to the offshore observations and seeing how those compare and whether the conclusions that we found from the first part hold for the second. So the data that I'm using here is coming from the Northeast US Shelf Long-Term Ecological Research Site, or the NESLTER. That's indicated by the star among the LTER network here. We have our nice broad continental shelf on the East Coast. And in particular, I'll be focusing on this region south of Cape Cod. And in this location, the dominant environmental variable is temperature. And we have our regular seasonal fluctuation in temperature, warm summers, cold winters. And we also have this spatial gradient that kind of mirrors that, where we have our colder, fresher water on the shelf mixing with the saltier, warmer Gulf Stream water. And the sampling plan of the LTER is designed to capture both of these sources of variability. We have the Coastal Observatory, which we heard about a bunch yesterday, MVCO, indicated by the star on the map on the right. And that red dotted line is our main cruise transect, which is capturing the spatial variability. All right, so here's MVCO, the offshore tower again. We heard about the imaging flow cytometer, but the data I'm using is coming from our more traditional flow cytometer, also deployed underwater at this station year-round. It's recording information about particles in the water, fluorescence and size, scattering. And here's just an example of how we can use that to cluster different groups. Synechococcus is shown in red, the Pico-eukaryotes are in green. And we put those instruments out there for a long time, they get covered in algae. We swap them out with different ones, and so then we can have this continuous time series of observations of the Picophytoplankton. And we can use that to get answers to our questions. So in particular, for seasonal environmental variability, what do we know about the Picophytoplankton response? Here's Synechococcus concentration against temperature, and it's on a log scale. And we'll go through the seasonal cycle, which is indicated by the colors. So starting with the early spring in dark blue at the bottom, we have low concentrations of Synechococcus. They undergo a dramatic bloom, reaching maximum concentrations in the early summer. And then there's a more gradual decrease in concentration back to winter lows. And this is a really nice pattern. These are daily averages. But it doesn't give us much mechanistic information about changes in the population. So as we know, changes in population size are going to result from both adding and removing individuals. In this case, it's primarily through division and mortality or loss. It could also be from advection, but we're going to focus on these for now. In particular, division rate is our term of interest, our growth rate, because that gives us some physiological information about the cells. If they're growing very fast, we understand that they're relatively happy in their environment. And if they're growing if they're dividing less, then there might be something limiting their growth. And as well, quantifying the division rate, we'll be able to infer then the loss rate based on the changes in concentration. In particular, for the Picophytoplankton, these processes of division and loss are happening at a much finer scale than this seasonal cycle, on the order of hours to days. So we're going to zoom in. This is just a week of data. These are eukaryote concentrations at MPCO. And again, we won't be able to see division or loss from just the changes in concentration, but that's not all the data that we have. We also have observations of cell size. So these are, the heat map is showing the changes in the population size over the course of the week. And we see this really nice diel pattern where the cells are getting larger and smaller. And the vertical bars here are indicating dawn, which we know from looking at the light data, which is also collected at the observatory. So the cells tend to get larger while the sun is nice and bright, and then they get smaller primarily through division. So this information has embedded in it the division rate that we're after, and we just need to figure out how to extract that information. We can do that using a size-structured matrix model. We bin all of the cells into bins by their volume, and for every time step, we allow those cells to either grow into the next volume class or divide and reproduce 2 cells of half the volume. So the transitions are shown on that matrix on the right, and the values of those positions will be informed by our parameters. And we make 2 assumptions, the first being that cells will grow more while sunlight is out, while there's brighter sunlight, and they will divide more once they get bigger. So we use the cell size observations and the sunlight data in order to fit the model parameters for this model to a given day. And then we run a simulation of the model, and we look at how frequently the cells in the model are dividing, and that becomes our estimate of the division rate for the cells in the water. And we think this works. So not only is the model able to reproduce the diel dynamics that we observe in the water at MPCO on a variety of days, it also produces division rate estimates that agree well with those that we would get from a traditional dilution series method. So this is showing our division rate estimates as opposed to the dilution series method for a variety of Synechococcus cultures, samples in the dilution series. So importantly, our method doesn't require taking the cells out of their natural habitat, and it also doesn't depend at all on cell concentration, because we're just using the cell size distribution. So we've done this for all of the observations that we've had every day of the time series for 2 different kinds of Picophytoplankton, both the Synechococcus and the Picoeukaryotes. And what has that taught us about the division rate over the season? So here, Synechococcus division rate is plotted on the top, and Picoeukaryote division rate is on the bottom, both against temperature again, with the seasons indicated by the color. So we see they have very similar responses, where both are showing an increase in division rate with increases in temperature in the spring, reaching maximum division rates in the summer, and then declining again. But importantly, the division rates that they're seeing in the fall are lower than they are in the spring for days of the same temperature. These are a seasonal climatology, I should have said that. So this suggests that they're not temperature limited in the fall because they are capable of dividing more at the same temperatures, but we aren't seeing that response. If instead we plot the division rate against the radiation, we can see that there's a nice linear relationship in the fall between the sunlight and the division rate. So as the sunlight is decreasing in the fall, the cells are dividing less frequently. All right, so that's our main conclusion from the stationary observatory. The cells are, the Picophytoplankton, both groups, are temperature limited in the spring and light limited in the fall. Is that the same response that we see offshore, or is this just a single location? So we're going to move on to the second part. The underway data that I'm looking at is coming mostly from the LTER cruises on the Endeavour, but we also have sampled opportunistically on other cruises where we're able to send the instrument. This instrument is the attuned flow cytometer. It's making the same kinds of measurements, but in this case, it stays nice and dry in the lab, and we have the water pumped from beneath the ship, so they're surface samples that are taken into the lab and sampled. In this case, every 2 to 5 minutes, because the ship is moving, we're trying to get as many samples as we possibly can. This is as fast as the attune will go. And, well, not to make appropriate res measurements, I'm sorry. And then on the left here, you can see that we can cover a larger spatial area. These are all of the cruise tracks of the 17 cruises in this work. And on the right, just a sense of the timing of when those cruises are. So colored by their season, we have the last 4 years. And you can see that we have cruises at all different seasons, but of course, each cruise is much shorter than our observatory data is. It's only about a week long, up to about 3 weeks. And so we have an average coverage of about 20 days a year. So just as a reminder, again, this will be including both the temporal variability that comes from the seasons and some spatial variability as we go offshore. And this is a nice clean pattern of kind of the general trend in the region. But of course, our reality is not so neat. We have just some satellite images to show you that there are fine scale features and that things can change rapidly from day to day in a cruise. But we can still see some general trends. Here we're looking at, in blue, the Synechococcus abundance binned by latitudinal bins, and in red, just the average temperature for all the samples. Note here that the sampling is uneven between the seasons because of the different number of cruises, and it's also going to be uneven across the latitudinal bins because it's just going wherever the cruise went, and some of these were opportunistic cruises. But in general, we have higher concentrations of Synechococcus offshore in the winter than we do near shore, and warmer waters offshore. And that relationship is more dramatic in the spring, where offshore concentrations of Synechococcus become even higher. In the summer, note that the bottom two panels have a higher y-axis because that summer bloom near shore is so dramatic. And we see lower concentrations offshore. And in the fall, there's no real trend here in Synechococcus concentration. So that was all concentration. And again, we're going to be more interested in the division rate, and that's going to help us interpret those changes in concentration. So here's where that panorama anecdote comes in, because we have our observations and the ship is moving, and we'd like to look at diel patterns, but we don't know that we're necessarily observing cells that have experienced the same environment, or most likely they haven't. So there's going to be some changes over the course of the panorama. At the top, we can see the concentration can change pretty dramatically over the cruise, but we still have this nice diel pattern in cell size over the course of the day. So we could go ahead and fit the model to the days starting at dawn, like we do at MVCO. But I would worry, and you might worry, that this would be sensitive to where we were in the water and whether we move between transition between bodies of water towards the end of the day or at the beginning of the day. And we wouldn't want to be subject to that kind of serendipity. So instead, we use a sliding window where we fit the model to each 24-hour period of the cruise, starting at every hour of the cruise. So we're going to have some overlapping data informing the model here, but we're going to end up with a sort of continuous estimate of division rate that can move with the cruise observations. And we can plot that against things like the average position of the data on that day, or the average temperature or salinity. And we can also look at the variation in temperature or salinity as an indicator of whether this was a reasonable observation of a given population. All right, so what do we see? Do we see the same response offshore as we do MVCO? So as a reminder, this was our nice seasonal pattern in Synechococcus concentration at the Coastal Observatory. And here is all of the samples from all the cruises that I've considered across the whole shelf. And so this is combining both seasonality and spatial environmental heterogeneity. First order, this looks great. So the seasonal response of Picophytoplankton seems to be the same further offshore. MVCO is not just a random, unique point, and that's part of why we put it there, is we thought that it would be representative of the wider shelf, and we're happy to see that it is. If we zoom in on a cruise that has a particularly wide range of temperature, like this one here, we can see that the response of Picophytoplankton in abundance is comparable, even if it's sampled over just a few days. So this is not looking at temporal variability, it's only the spatial change in temperature, and it's following the same response as we see over time at MVCO. So we can kind of have this space for time substitute. Again, looking at all the data together, it seems like the pattern holds about until those temperatures, those very high temperatures, which we never see at MVCO. So that could be that there's just totally different water out there, right? We have warm core rings and things, and those concentrations are lower than we would predict based on the observations at MVCO. But I'll come back to that. Now we're going to move on to the division rate response. So you remember our nice relationships between division rate and temperature and division rate and sunlight. These are just Picophytoplankton. These are the annual climatologies. These are the daily data that comes from. So this is showing the whole variability in our time series. And I do this here because I think it's more comparable to the cruise data because we don't have the extensive time series that we do to average and get those climatologies. So now on the right is the cruise data. All of our models fit to all those sliding window days. And in the gray shaded behind that is just the same data that's on the left, so you can more directly compare. So the division rate is following a similar response seasonally, where it's increasing as water's warm. But the division rates are lower than those that we see at MVCO at similar temperatures. So again, we're seeing this gap where they're not able to attain those maximum rates that we saw at MVCO at the same temperature, particularly in spring, where we saw those highest rates. This could suggest that the cells are limited by something other than temperature in this area. It could also be that there's some compositional variability, and the cells offshore are just taxonomically different than those near shore, because Picophytoplankton is a rather large group. Relating to sunlight, and in the summer and fall, we see that the relationship is more similar to that MVCO, where sunlight is decreasing and those division rates are decreasing as well. And here's just the average version of that data, so it's a little bit cleaner to look at. But again, we're seeing a response to temperature that looks more like the fall at MVCO, where they're light limited. One hypothesis is that they actually are light limited offshore. It could be that there's more mixing offshore where the waters are deeper. And even if the waters are warm, they're not stratified enough that they're getting the light they need to attain their maximum rates. So this is a hypothesis I hope to explore by looking at mixing over the course of these cruises. And I just wanted to end with this plot, which is showing the same data where we have our cruise division rates in color on top of the MVCO division rates in gray over time for days of the year. So again, because the waters are warmer offshore in the winter, we're seeing higher division rates in the winter offshore than near shore. But the increase in division rate that we usually see in the spring is not as dramatic offshore as it is near shore. And I'm proposing that this is because they're light limited due to mixing offshore, and that they're able to attain those higher growth rates later once the water is stratified. And I think this might help us explain some of the anomaly in our concentration temperature relationship, because this region where we have lower concentrations than expected corresponds to the region that we also have lower division rates than expected. And I just want to highlight that our division rate estimates do not take concentration into account. So there's no reason that just seeing lower concentrations would give us lower division rate estimates. But there is biologically a reason why lower division rate would lead to lower concentrations if loss rates are similar. And loss rates could be similar if they're temperature driven in the spring. So I'll end with my conclusions here, that our continuous observations of cell traits are providing mechanistic information about changes in picoplankton abundance. The relationship between temperature and picoplankton abundance is consistent across seasonal and spatial gradients, and it's consistent from MVCO to offshore. And I have a new spatially explicit hypothesis to continue exploring for the controls on sim growth, mainly being that though they're temperature limited at MVCO in the winter and spring, they might be more light or mixing limited in the winter and spring offshore, and then they're only able to attain their maximum growth rates in the summer. With that, thank you all for listening, and thanks to our funders.
Thank you, Bethany, and another great data set. Wonderful to be able to get those growth rates in situ, and amazing. Questions?
Thank you. Very interesting. Presumably on those cruises, there's sort of genomic data being collected. What have you learned about how your data interfaces with, the presence and absence of different clades of Synechococcus?
So at MVCO, we know that there's one strain that's dominant for most of the year. So a lot of the inferences we can make from the model, we think are physiological responses of a single clade. The cruise data we haven't yet looked at, but we certainly plan to see if there are changes either over space or seasonally offshore. Yeah.
Sort of as an add-on to that, so for other pico-eukaryotes outside of Synechococcus, do you have an idea of what the range is, like in the flow cytometry when you're sort of detecting these organisms, what sort of that taxonomic range is between organisms that you're measuring?
I'm not sure I understand the question.
Yeah, so in flow cytometry, I guess when you're taking these environmental samples, how much are you able to sort of differentiate, like if you have different organisms, like taxonomically different organisms?
Oh, just within the flow cytometry?
Yeah.
It's really mostly breaking it down into Synechococcus and the pico-eukaryotes and Prochlorococcus if we saw any, which we don't in this region. Sometimes you can see, even in the example I gave, there are like 2 little clusters of pico-eukaryotes. And so we don't know what they are necessarily, but it can be a bimodal distribution in size. And so that suggests that there is at least some variability. And our model does account for that. We allow for 2 populations to account for those bimodal days.
Very nice data. It's the type of data that many of us aspire to get. It's really incredible and interesting to link abundance and growth. I was wondering, the paper that you put up from our group in the beginning, the Flumbaum et al., they hypothesized that the abundance of Synechococcus declines around 20 degrees due to competition with Prochlorococcus. So I was wondering about whether you see any of that in sort of the offshore transects when Prochlorococcus starts to come in. Does that suppress the abundance of Synechococcus?
Okay, I think I only heard parts of that.
I can repeat it again. The point is just that in that paper we hypothesized that the abundance of Synechococcus above sort of about 20 degrees when Prochlorococcus really starts to become abundant is suppressed by the competition because Prochlorococcus is smaller and takes up nutrients to a lower level. So Synechococcus has to rely on sort of alternative forms. And so I was wondering about whether in your offshore transects, do you see sort of a similar pattern whereby when Prochlorococcus starts to become abundant, that's when you start to see a suppression in abundance of Synechococcus.
Okay, right. So the hypothesis would be that if we go further offshore where there's more nutrient limitation, we would see more Prochlorococcus and less Synechococcus. I haven't looked at it, but that would be interesting to explore. Heidi, do you have an answer?
Heidi says they haven't gone far enough yet to be able to get that that feature. All right, we've got an online. Is that Ken? Ken, go ahead.
Yes, thanks. Very impressive, in particular that algorithm for getting the growth rate. One thing I would be particularly interested in would also to be then to see the actual loss rate, so you can see the changes in predation. But my question was rather part of that algorithm assumes that growth is driven solely by light. Have you thought about whether that is correct and whether there are some biases introduced there?
Okay, I think I'll say two things in response. The first being that, yes, we can infer loss rate, and that is really exciting. At MVCO, it seems like a more reasonable step because we can smooth the concentrations over time and get rid of tidal effects and things. So we have done that there. Haven't done it for the cruise data because we don't want to just take the concentration at the end of the day and at the beginning of the day and compare them to get a loss rate because it might be very sensitive to exactly when those beginning and end of the days are. But I do plan to do some of that. And then the second piece is that our model assumes that growth is dependent on light. It certainly does. And there are plenty of assumptions that other people have explored, making the model more complicated. Yeah, we think that it works pretty well, validating with the dilution series estimate. One thing that comes up a lot is nutrients, and we can, again, look at the relationship between the growth rate that we estimate and the nutrients that are available, but whether the actual growth rates that we're comparing are biased based on not considering nutrients to start. I hadn't considered. We would need nutrient observations at a much higher resolution than we have to incorporate them into the model, or at least estimates of them. So, interesting. Thank you.
One more question.
Regarding the moving window that you're using to try to account for the fact that you're moving, when you actually looked at it, maybe you don't remember, did actually just going over the 24-hour or light day solve the problem, or did you really need that moving window kind of thing to account for the moving? In other words, could you ignore the fact you were moving, or no, it was important to account for your motion, so to speak?
So there are definitely days that we have to quality control out because there are discontinuities in the size distribution between traveling through bodies of water. So it's possible that I could have just looked for those and removed them. But I was really just worried about the subjectivity of picking a particular set of windows and only getting 4 estimates for a cruise. These are week-long cruises mostly. So the sliding window is not really accounting for the fact that we're moving. It's just trying to reduce some of the uncertainty that comes from where we choose our windows.
Great. Well, thank you very much.